Welcome to Computational Biology. Today, we are diving into a crucial topic, sequence submission tools. Imagine you have just recorded a fantastic video and you want the whole world to see it. You do not just throw the raw video file onto the internet. You go to a platform like YouTube, upload the file, and carefully add a title, a description, and relevant tags. In the world of computational biology, when a scientist discovers a new DNA or protein sequence, they must do the exact same thing to share it with the global scientific community. Let us explore how this works. Building on that YouTube analogy, let us look at what these tools actually do. Sequence submission tools are specialized software platforms that allow researchers to send their biological discoveries to massive public databases. Why does this matter? Because raw biological data on its own is just a string of letters. It needs context. These tools ensure that your raw biological data is packaged neatly with all the necessary background information so that anyone in the world can find it and understand it. They act as the bridge between an isolated laboratory discovery and the global scientific community. Now that we understand the basic idea, let us define these tools formally. Sequence submission tools are specialized software interfaces designed to accept, validate, and format raw biological sequence data, along with its associated metadata. They do not just accept any file, they act as strict gatekeepers. These tools ensure that every piece of submitted data strictly adheres to the formatting rules set by the International Nucleotide Sequence Database Collaboration, often abbreviated as INSDC. This rigorous validation process is what keeps our global biological databases organized, reliable, and scientifically useful for everyone. We just mentioned the term metadata, so let us explore exactly what that includes. Metadata is essentially data about your data. In computational biology, metadata includes crucial background details, such as the name of the organism, the specific gene name, the scientists who discovered it, and the geographic location where the sample was found. Without this descriptive text, a DNA sequence is virtually meaningless to other researchers. By attaching rich metadata, scientists provide the biological context necessary for others to verify their work, conduct comparative studies, and build upon their initial discoveries. Knowing what these tools do, let us look at a couple of specific examples used in the field. There are different tools designed for different scales of work. For instance, Bankit is a simple web-based tool that is perfect for small, routine submissions, like a single gene. On the other hand, Sequin is a robust standalone program used for highly complex or massive sequence submissions, such as an entire genome. Researchers must choose the appropriate tool based on the digital size and biological complexity of the data they are trying to upload. Once you have chosen your tool and submitted your data, what happens next? Once the tool verifies that the data is biologically logical and correctly formatted, it issues a unique alphanumeric code called an accession number. You can think of this accession number as a permanent, globally recognized barcode for that specific sequence. It is the ultimate identifier. Whenever a scientist publishes a paper, they include this number so that anyone reading the research can instantly retrieve the exact genetic sequence from the public database and analyze it themselves. With the mechanics clear, let us discuss why these sequence submission tools matter so much to modern science. Without them, the vast amount of biological data generated every day would be trapped on the personal computers of individual scientists. These tools democratize science by creating a standardized, searchable, and globally accessible library of life. For example, during the outbreak of a novel virus, scientists rely on these tools to rapidly share the viral genome with the rest of the world. This immediate sharing allows researchers to develop diagnostic tests and life-saving vaccines in days rather than years. Understanding the importance of these tools leads us to a practical question. How do we manage the digital footprint of this data? While sequence submission is primarily a data management process, computational biologists must carefully consider the digital size of the data they are submitting. We can represent the total digital footprint using a simple linear equation. S equals n times b plus m. Here, S represents the total submission file size in bytes. N is the total number of biological residues, like nucleotides. B is the bytes required to store a single residue, typically 1 byte. Finally, M represents the size of the metadata in bytes. If the sequence length increases, the total file size increases proportionally. Now that we have our formula, let us see this with a concrete example. A researcher has sequenced a small insulin gene from a newly discovered mouse species and wants to submit it using Bankit. The gene is exactly 1,500 nucleotides long, and the metadata takes up exactly 2,000 bytes. Assuming 1 byte per nucleotide, we need to calculate the total file size. We substitute n equals 1,500, b equals 1, and m equals 2,000 into our equation. Multiplying the sequence length by the byte size gives us 1,500 bytes. Adding the 2,000 bytes of metadata yields a final size of 3,500 bytes. Because this file is so small, a simple web-based tool like Bankit is perfect. Having seen a small submission, let us apply the same equation to a much larger dataset. A team has completely sequenced the genome of a novel E. coli strain. The genome is 4 million base pairs long, and the extensive metadata amounts to 50,000 bytes. We substitute n equals 4 million, b equals 1, and m equals 50,000 into our formula. The raw sequence size is 4 million bytes. Adding the heavy metadata overhead gives us a final footprint of 4,050,000 bytes, or roughly 4.05 megabytes. While small for a modern computer, parsing millions of bases simultaneously might crash a standard web browser. Therefore, researchers would bypass web tools and use robust standalone software, like Sequin, for this job. Now that we have calculated submission sizes, let us visualize the entire process we have been discussing. On the screen is a workflow diagram showing the journey of biological data. Starting on the left, we have our raw sequence data, combined with its metadata. This package moves into the submission tool, like Bankit or Sequin, represented by the computer monitor. Here, the critical steps of validation and formatting occur. Once the tool confirms the data is biologically logical and correctly formatted, it is sent to the INSDC public database, shown as the blue cylinder. Finally, the database generates and assigns the unique accession number, represented by the document on the far right, anchoring the discovery permanently. We have covered the full topic of sequence submission tools, so let us reinforce the most important ideas. First, these tools are the critical bridges that connect isolated laboratory discoveries to the global scientific community. Second, by enforcing strict formatting rules and validating metadata, they ensure that public databases remain organized and reliable. Third, they transform raw, unformatted strings of biological letters into permanent, globally accessible knowledge, anchored by unique accession numbers. Finally, understanding the digital size of your submission is key to choosing the right tool for the job. Mastering this standardized process is a fundamental skill, as the collaborative nature of modern bioinformatics depends entirely upon it.